Okay guys, next up we're going to look at how we make cells, okay? And one specific type of cell reproduction. And I know most of you have heard it before, because if you hadn't heard it with me, you probably heard it with Miss Carol down in 7th grade when you took life science. We want to talk about mitosis and the cell cycle, okay? Your body is made up of trillions of cells, and each one of those cells is eventually going to make new cells. Well, how does it do that? Well, every living cell has a life cycle. And there's two major sections to your life cycle. The first stage is called interphase. Now, again, if you remember this way back when, interphase is what the cell is doing most of the time. It's living its life in interphase, okay? The cell is growing, you can see the nucleus, there are no chromosomes that you can see because they're not condensed up. And it doesn't look like anything's really happening when you're looking at it just under a microscope. It's kind of boring looking because it looks like it's just kind of resting. And they used to call it the resting stage. But the cell is not resting. It's growing and doing its own thing. It's making mitochondria, is making ATP, ribosomes are making proteins, the Golgi, and everything's doing its own job. You just don't see any differences really. Okay? And again, that's interphase. There are 4 major stages of interphase. Actually, there's usually 3 of them, but there is a 4th one that's kind of an oddball, okay? The 4 stages are the G1 phase. This is where the cell is just growing. The first growth phase, G1. It's getting bigger, just like every living thing does. It gets bigger and bigger and bigger, all right? The S phase is where we get to a certain size and we're like, okay, we've gotten big enough. We need to split because cells can only get so big anyway. So we're going to split the cell. Well, if we're going to make 2 cells, we got to start copying DNA replication, right? We got to copy the DNA. This occurs in the S phase. Okay, and this occurs relatively quickly. Doesn't take that long. You think it would as big as DNA is, but it really doesn't take that long to copy all of the DNA. And then the last phase, normally, is something called the G2 phase. This is the secondary growth stage. Basically, the cell gets a little bit bigger. It gets all the organelles like the mitochondria, right? It gets everything to where that when we get ready to split, we'll have 2 sets of cells for everybody and it won't be a problem, okay? That's basically what we're doing. Now this G0 here, right here, nothing's really happening in G0. A lot of times some cells will go into this stage where they won't grow and reproduce new cells, all right? One of the main places in human beings that happens a lot is in the brain. Brain cells tend to hang out in G0. They don't get any bigger, and they don't, I mean, they're still living their life. They're still doing all the proteins and the ATP. They're still doing stuff. It's just they're not going to get ready to get to a point where they go through S phase and G2. They're not going to do that. They're just kind of hanging out, which means, again, if you damage brain cells or nerve cells, they don't reproduce themselves very often. So that's permanent kind of damage there, okay? So that is, again, and I know it's a little fast, but that's basically the cell cycle as far as interphase, right? And a cell spends most of its life in interphase, probably 80 to 90%, maybe even 95% in certain types of cells, of its life in the whole set of interphase, okay? So that's a pretty long stage. And most of the time when you look under a microscope at a cell, you're going to see it in interphase. Could you see it in mitosis phases? Yeah, it's harder to see because that doesn't take up much of its life cycle. Okay. Now, with mitosis, this is where we're going to divide the nucleus. Always remember, mitosis is the division of the nucleus, nuclear division. Okay? Now, and again, there are 4 stages, and we've heard these stages before, haven't we? There's prophase, which is the first stage, right here. What happens? The nucleus and the nucleolus start to break down. So what we're having is the nucleus is basically falling apart. The DNA coils up into the chromosomes. These little things called spindle fibers, which are little microtubules, start to grow. By the time we get to metaphase, the chromosomes are lined up. See here, they're lined up in the center of the cell. They've got these microtubules, these spindle fibers grabbing a hold of them. So they're literally just lining up like you used to in elementary school, lining up in the center and, you know, lining up in a line. Make sure you're all lined up perfectly. Okay? Anaphase, the spindle fibers are going to pull them apart and what you'll have, remember we copied each one, so each one of those was called a sister chromatid. We literally rip those in half, and we get pulled towards the opposite side of the cell. And then the last stage is telophase. Everything goes in reverse as prophase. The nuclei, because they're making two cells, two nuclei form. Nucleoli, I hate to say nucleolysis, but you get the idea. Two nucleoli start to form, and the DNA, or the chromosomes, uncoil back into the DNA structure. Okay? So literally, telophase and prophase are basically reverses of each other. But the whole purpose is, we get these copied chromosomes and we split them. And I'm going to show you again. See right here? There's light blue, orange, dark blue, purple, red, and then they split, and you got one of each set. Here's my copy, here's your copy. Then we reform the two nuclei. Okay? And we all got a copy of everything. So this guy's going to be identical, this guy's going to be identical. Okay? Now, the last stage of a cell cycle is something called cytokinesis, which is basically we're splitting the cell into two cells. Okay? That's all we're doing. We're creating two new identical daughter cells. And the thing is, you already figured this out. Plants and animal cells are not the same. So you're actually going to have some differences here. One of the things is, with an animal cell, they've got some fibers in here that basically start to pinch the center. It's called a cleavage furrow. And eventually this thing will pinch all the way through. Next thing you know, they're two separate cells. Okay? Plant cells can't do that because they have that cell wall. So what they'll do is they'll start to grow what we call a cell plate in the center. And that plate will eventually come right here, and it will separate both sides so that now you have a cell here and a cell here. So we have to grow a new wall, basically what we call a cell plate, in between them until they completely separate from each other. Here we just pinch it and then split. That's it. Okay? But again, that's how we're making two identical cells through this whole cell cycle, right? Okay. Now again, when we're talking about who controls this, always remember, there are growth factors and proteins that regulate this. Now, what is that? Why is that a big deal? We're going to see in a minute, okay? And every once in a while we have these things called checkpoints, making sure that the cell cycle is going the way it's supposed to. Because you can already probably figure if the cell reproductive cycle doesn't work right, we're going to run into problems, okay? So, metaphase, for example, make sure all the chromosomes are lined up and the spindle fibers attached correctly. Now, does that always work? No. Well, what happens if these checkpoints and this control doesn't work properly? What do you think we're going to start getting? Well, you should already kind of figure it out pretty quickly. We're going to get something called cancer. Cancer is simply defined as uncontrolled cellular growth. Okay? And again, there are other things that may lead to this. You can see this right here. Exposure to radiation, smoke, sunlight, viruses. There's all kinds of things that can cause cancer because cancers are basically mutations where the cells grow and grow and grow. There's no check on them. So you end up growing these huge amounts of cells that aren't working properly. They're not acting right. Okay? And that's a bad thing. And again, you hear all your life that people talk about, well, maybe they'll find a cure for cancer one day. Well, cancer is nothing more than mutation. They may be able to find cures for them, and once you get it, they can give you a treatment that will stop it. Right now, the two main treatments that we have are chemotherapy and radiation. What do? Well, if it's a tumor, we can spotlight it with chemotherapy, radiation. If it's a tumor, we can point radiation to hit right there at the tumor and kill that tumor, hopefully. If it's not a specific tumor and it's kind of spread, we run through something called chemotherapy. We inject you with drugs, chemicals that hopefully will kill the cancer cells, but not kill the healthy cells. And again, you've all seen people who've taken chemotherapy. It's really difficult. Both of them, radiation and chemotherapy, because it does kill living, healthy cells as well. What we're hoping is it kills all the bad ones first before it kills all the good ones. That's what they're hoping anyway, okay? And again, we're always looking for new medications that are more specific, that will only target specific cells. And that's what they're always working towards, and maybe one day they'll figure it out where they can treat most cancers and pretty much stop most cancers from developing past a certain point. We hope anyway. We never know. But again, hopefully they will. Now, so that is the cell reproduction of most cells in your body, right? That is your somatic cells, mitosis, right? The next video we're going to look at in just a second is dealing with how cells reproduce to produce gametes. So you'll see that one next up, okay?